Well, thank you so much. It's awesome to be here. So it's nice to see some old friends and meet some new friends and from all over. I actually did my medical and graduate school here on this campus, and so it's nice to be back. And it's congratulations, Dr. Beam, on a great forum. It's been I think so far it's been great, and then the rest of the program looks equally good. So in the CME world, I have no disclosures. But I do have a caveat, right? And so I think Dr. Beam probably more eloquently outlined the caveat earlier. But when you think of a geneticist view of the world, so I do research in human genetics, the geneticist view of the world, of course, is biased. And so the view is that you have a genotype, and then there's this like black box, right, which is the rest of biology. And then, of course, you have a phenotype. And there are very clearly behaviors and phenotypes that cannot be simply explained by genotype. And I won't get into the black box. I'll just sort of focus on one of the inputs to that black box, which is genotype. And in my space, so in the cardiovascular world, we sort of lump things into two broad categories, right? So we have Mendelian forms of disease. These are diseases that segregate within families, in which the phenotypic variance can be explained by a single allele of a large effect. And so there's cardiomyopathies, which are heart muscle disorders. There's arrhythmia syndromes, which are electrical problems. There's lipid disorders, disorders of cholesterol metabolism, in which cholesterol builds up at very high levels, alternatively also very low levels, depending on the syndrome. And then there's vascular syndromes, and I'll talk about one of these vascular syndromes today, where people have aortic disease and also can be peripheral disease as well. Most human diseases and most human traits are, of course, not Mendelian. I think Matt nicely laid out sort of the complexities with mapping genes and populations. But most human diseases and traits are under the effect of many genes. And we know that they're heritable, right, because there's clustering within families. And of course, you can do formal heritability calculations to figure this out. But the clustering within families does not follow the patterns of Mendelian segregation. And so although there's genetic factors that underlie the risk of disease, it's not due to a single gene. And so we call these disorders polygenic. And a disorder that we study a lot in the lab is coronary disease. And so there's been lots of population mapping for coronary disease. Of course, blood pressure is normally distributed in the population. Lipids are essentially normally distributed in the population. And this goes, of course, on and on for diseases and traits within this space. And I'll tell sort of two stories today. One will be on the sort of single gene side, and one will be on the common disease side. And a goal, not necessarily the entire goal, but a goal, of course, of precision medicine, and I think what we're all talking about today, is how do we incorporate genetic information into clinical practice in both of these realms. So you take a family that presents to the clinic, and then you take an individual that presents to the clinic, and how do you use genetic information to inform both of those clinical practice arenas. And I don't necessarily, I'm not a time traveler, I don't bring news from the future, but precision genetics is already in practice, right? And this has been in practice for a long time. And I highlight this just to say that when I talk to my division chief or when I talk to other people around campus, they think that this is some far-off futuristic idea like the movie Gattaca, right, where we have everybody's genome sequence and we can predict what career they're going to have in the future. But of course, in both single genes and complex genetic situations, we've already been practicing this for quite some time. And so, of course, we don't give blood without understanding what somebody's blood type is. And of course, that's a surrogate for a genetic marker of a single locus that informs therapy, right? We choose therapies based on that genetic information. We don't necessarily genotype people, but we have a surrogate of their genotype. So we choose therapies, and we avoid putting people at risk based on genetic knowledge. And one thing that you learn in the first year of medical school is that you take a family history. And we use that in cardiology practice all the time. You want to know what somebody's family history is because that informs how you think of their risk. Of course, these are sort of very blunt tools, and I'll try and convince you today that we can do better in both of these arenas. We'd like to be, of course, more precise when informing decisions for both monogenic and polygenic disease. And I'll start with the story of a 35-year-old gentleman that came in with aortic dissection. So on the top panel is a CT scan. And for those of you that don't spend a lot of time looking at these, this is a transverse section of the chest. And so the spine is on the bottom, individuals lying on the table. So the spine is here, the sternum's here. And the aorta, of course, is the main blood vessel that leaves the heart. It goes over the aortic arch, and so you have a part that's ascending, leaving the heart, and then you have a part that's descending. And shown here is the descending aorta. So that little white circle next to the spine is of normal caliber. So that's what the aorta should look like. And then, of course, over here is the ascending aorta. So as it's leaving the heart, this is obviously quite large. And you should not see a line in the middle of the aorta. And so that's actually the aortic wall, which has separated. So blood is flowing into the wall. This is that dissection flap. And this is, the aorta is basically unraveling in this patient. And of course, this is a medical emergency. Fortunately, this person survived, so they made it to the operating room. They had an aortic root repair. And this is the histology, so shown here. Histopathology showing sort of typical changes, which include cystic medial necrosis. So we sort of see this in most people that come into the clinic or most people that come into the hospital with this condition, not really giving us insight into what this person might have. So the pedigree is, of course, quite interesting. So shown in the blue arrow shows the proband. And you can see that he had a mother that ended up having dissection, and then actually had two first cousins who also had aortic disease at an early age. The aunt, which we can talk about later, the aunt who's an obligate carrier did not necessarily have an overt phenotype. And again, we can talk about that later about what that might mean. But we thought this was a reasonable family for an autosomal dominant form of aortic dissection. And there's gene panels. So we heard this morning about gene panels and the utility for gene panels. And of course, there's gene panels for this syndrome. This is a known syndrome. And this individual underwent clinical gene sequencing for one of these gene panels, and they failed to identify a causal mutation. And so we said, well, this is a nice family. Maybe we can actually learn something about this disease. It's a small family, however, and so we couldn't use the traditional methods of linkage because it's very unlikely that, that linkage in a small family like this would lead to a significant LOD score. And so we used an approach of doing direct genome sequencing. So we took this proband and his first cousin, and we did whole genome sequencing to try and see if we could map a gene. As we discussed earlier, there's 3 billion base pairs in the genome, and of course, when you do whole genome sequencing, you get 3 billion possibilities. And so, of course, you have to then sort of go through the haystack of needles to try and find the needle you're looking for. And I'll tell you sort of our general approach, which is not necessarily incredibly novel, and a lot of labs are doing the similar things. Of course, the first thing to do is to identify variation that co-segregates with a phenotype. And so we have this idea that this is autosomal dominant. And of course, we require that both the proband and his first cousin carry what we think might be the disease-causing allele. And as we think that most Mendelian disorders are caused by variation that significantly alters an encoded protein, and so that's the next thing that we sort of look for. And then we have a host of families around. So we've done this for a variety of families with a variety of disorders. This is a pedigree of a dyslipidemia family, so very high cholesterol. And as long as you don't think that the same mutation might be causing two different phenotypes, which of course there are examples that it does, if you think the phenotype is sufficiently unrelated, you can actually leverage those sequence cases locally to eliminate variation that you find in the family in which you're trying to solve. And this is most typically sort of most useful for sequencing specific errors or sequencing specific artifacts that the sequencing center that you're using might produce. We heard earlier today about population databases, and so this is now we're in the order of hundreds of thousands of exomes that are available. And so in the coding space, we actually have a very good idea in certain populations, and most in Europeans, we have a very good idea about what is the population frequency that with which we find alleles. And so, of course, if you think that the disorder that you're trying to solve is rare, and you think that there's a reasonable penetrance, you can actually work out what you think the upper bound of the frequency of the causal variant might be. And so you can sort of look for things in the population that are very rare. And then you're sort of almost always down to a handful of things, and now you can genotype in the remaining pedigree. So you can You have a cousin over here who's affected, and you can ask that, that person carry it. You have a brother who's unaffected, and as long as you're convinced that the phenotype in that brother is real, then you can eliminate variation that's found in the brother. So to make a long story short, we identified a missense candidate mutation in a gene lysyl oxidase that we thought was the cause of disease. Lysyl oxidase was actually a great candidate gene. So it's an amine copper oxidase, a copper amine oxidase that cross-links elastin and collagen, which are the two main components in the aortic wall. And so we said, well, this is great. And then we looked in the literature, and we read a little bit more about the gene. And shown here is the missense mutation. So it's methionine at position 298 is replaced by arginine. And if you look at the multiple sequence alignment, so you can sort of take all of the species and you can align that fragment or that section of lysyl oxidase, and we like to joke in lab meeting, every organism that had an aorta had methionine at position 298, or the orthogonal position in that organism. And so it was a great candidate mutation, and we looked more in the literature, and we realized that there had never been a human that had been described with a mutation in this gene. So there was no human phenotype associated with variation in the gene. And so it was very difficult for us to claim that this was causal because we didn't know what variation in this gene would do. And it's also a missense change. And we know through population genetics that about 80% of up to 80% of missense changes are functionally neutral. So it's very difficult for us to claim that a missense mutation in a gene that had never been associated with disease in humans was causal. And so we set to sort of try and figure out how are we going to actually prove causality. We went through the we went through the freezer, and we found a bunch of other cases that we had collected, and we sequenced the same gene in those individuals and didn't find anybody with a mutation. And so I called up Bob Meekum, who's on the left here. So he's a vascular biologist at WashU, and then Vivian Lee, who's an incredibly talented grad student. The three of us sort of set out to show that this was a causal mutation, and this was published last year. And this is a poor man's version of CRISPR-Cas9. I need to contact Dr. Beam for his AV group and the video that we saw earlier today. And so as that video showed, there's now an ability to use this CRISPR-Cas9 genome editing approach to specifically edit base pairs in the genome. And of course, the advantage of doing this is that you can take a mouse and you can, through this precision sort of CRISPR-Cas9 editing, you can generate a mouse that's normal. Of course, that's the mouse that's unedited. And then you can generate littermates that have the human mutation in the orthologous position. So they're otherwise genetically identical. It's just that they have a missense change that's been knocked in. So Vivian set out to characterize the mice. And it turned out that heterozygous knock-in mice were viable. And so she was somewhat disappointed, but not too disappointed because we kept on pushing. And so shown here are aorta from these mice. And Vivian has, before dissecting the heart out, she's actually injected dilute yellow latex. So it basically internally casts the aortic vasculature and all of the branch vessels. On the left are wild-type mice, and on the right are mice that are heterozygous for the human mutation. And you can see that grossly, the aortas look normal. Although when Vivian looked at the ascending aortic length, she actually noticed that it was slightly but significantly longer. So that sort of gave us an idea that although the gross sort of appearance of the aorta was normal, that perhaps there was actually some subtle abnormalities. So then Vivian again went on even further and took the aortas and did electron microscopy. And you can see on the upper panel, these are wild-type mice, and they have smooth elastic lamina. So you can, I hope, appreciate these are the elastic fibers that are in the wall. And these mice have these sort of contiguous, smooth elastic fibers. And mice that are heterozygous for the mutation in humans have highly fragmented aortic elastic fibers. And so these are sort of disarrayed, not contiguous, sort of not doing the structural work that they should be doing. And then Vivian, of course, then bred the mice to homozygosity and found that they were universally perinatal lethal. And you can, I hope, appreciate that compared to the wild-type mouse on the left, mice that are homozygous for the human mutation have tortuous aortas. They actually have ascending aortic aneurysms, sort of shown here and here, and then they actually have spontaneous aortic rupture, as evidenced by the blood clots there, which is likely the cause of death for all of these mice. Vivian then actually took protein that was being made by embryonic fibroblasts from these mice and did an activity assay. And I think you can appreciate that the activity of the homozygous mice that are producing the homozygous mutant protein basically have no lysolyl-oxidase activity, confirming indeed that this is a loss-of-function mutation. So with this one family, we now have a new diagnostic target, a new prognostic target, and we actually have a mouse model of the family. And so we can potentially test what we have, we can potentially test new therapeutic targets as well. So that's one side, right? That's sort of the families that come into the clinic, and how can we think about being more precise with genetic information in families? And then I'll tell you one story about how we've been thinking about doing this in the population. And the disease I'll talk about is coronary artery disease. So coronary artery disease is the leading cause of death in the United States and actually worldwide. It's caused by blockages in the heart artery that then leads to necrosis of the heart muscle. And it's an ideal target for prevention. So there's cardiac prevention clinics all over. And one of the reasons why it's an ideal target for prevention is that it is a disease that takes decades to progress. And so there's an asymptomatic phase over decades where plaque is building up in the heart arteries. And then eventually there's a plaque rupture and that causes a heart attack. And this is, unfortunately, we often treat people when they come in and they have advanced disease because that's when they come to clinical attention. And so this is when we start aggressive risk factor modification when they've already developed disease. And wouldn't it be better if we could identify individuals early who are likely to go on to develop disease and treat them early? The other reason why this is a great disease for prevention is that we actually have therapies that work. Unfortunately, most of the therapies that we have lower LDL cholesterol, and that's why they work, but they're quite effective at reducing risk of disease if started early enough. So there's been lots of debate in the popular literature. Some people say, well, we should just take statins and just put them in the drinking water. I mean, everybody should be on this, and maybe that's an approach. I think a different approach, of course, is to take populations and to identify specific individuals in those populations that might be at high risk and to ask, can we identify those people? And if we can, would they actually benefit from therapy? In terms of thinking about risk stratification, genetic factors, I think, have several unique properties. They represent fixed risk over your lifetime, right? So to the extent that at least we look at germline variation, these are inherited at birth. And they can be measured any time in life. So they can be measured when you're 20, 30, 40, and we can actually potentially use that information at that point to stratify your risk. As Matt alluded to earlier, in complex disease, coronary disease, there's been quite a bit of work, as well as in Parkinson's disease, looking at the inherited basis in populations. And through genome-wide association studies, there's now actually over 100 loci that robustly associate with disease. And only 40% of these actually map to known risk factors. And so for a young guy like me, that's kind of exciting because this means that there's potentially 60% of the loci are brand new pathways and brand new targets. Of course, this has been a long slog, and the hard work is just beginning in terms of actually figuring out what is the causal gene. And actually, the causal gene at most of those loci totally unknown, and we don't have a clue at those 60% of those loci what the causal gene is. And actually, I should also say I don't have a slide of the allele frequencies and the effect sizes, but it's also very similar to Parkinson's disease, where we have very common alleles, so allele frequency of 50%, meaning half the chromosomes in the population have what we call the risk allele, and it raises risk by 5% or 10%. And so can we actually use these, right? So do these genetic factors, even if we don't know the genes, and even if half the chromosomes in the population have the allele, and it only modifies your risk by 5 or 10%, can we actually use these to predict prospective risk in populations? And I'll tell you the story that we published 2 years ago, and hopefully convince you that indeed we can. And so in this study, we actually took a little under 50,000 participants of 4 statin therapy trials. And so these are individuals that underwent randomization to placebo or statin for primary prevention, or different types of statins for secondary prevention. And at the time, they had DNA and genetic consent obtained. And these are the titles of the 4 studies there. And we calculated what's called the polygenic risk score. And so at the time, there were only actually 27 markers that had been associated in the literature with heart attack. And the idea of course, is that you have, if these truly represent risk, and if they're truly additive, meaning they're independent, if you can have 0 of these alleles, or you can have 54, assuming that we're all diploid, which I think we are. And the idea is if they're truly additive, then of course the number of risk alleles should increase with risk. You can actually calculate this, and we did, and it turns out that this is normally distributed in the population, this genetic risk score. And we arbitrarily took the bottom 20%, and we said, well, those people are at low risk. We took the top 20%, we said those people are at high risk, and then we took the middle, and we said, well, they're sort of intermediate. And indeed, the genetic risk score stratified risk of disease in the arms of in the placebo arms. And so if you take the low risk group and you set them to be the reference, the intermediate group is about 34% increased risk, while the high group has a 72% increased risk of disease. And I should note that these analyses are actually all adjusted for traditional risk factors. So smoking, lipids, blood pressure, whatever you can throw into the model, this still stratifies risk. And not in this paper, but in other work, we've actually demonstrated that this is actually independent of family history. And so in individuals with a positive family history, this genetic risk score stratifies risk. And in individuals with a negative family history, it also stratifies risk. Okay, so we know that we can actually do this. You might think that, well, maybe that risk is not modifiable, right? So you have this genetic risk, and we just can't do anything about it because I told you that 60% of the pathways are not related to lipids or blood pressure. And that's basically how we treat people in the clinic. We say, stop smoking, we treat your blood pressure, and we lower your lipids. And so if you're at really high risk, can we actually do anything about it? Well, the nice thing about the data set that we worked with is that these are completed statin therapy trials. So I told you about the placebo arm, but we also actually genotyped everybody in the treatment arm as well. And this is the forest plot of the odds ratio, either favoring statin on the left or favoring control on the right. And you can see that for the low risk, intermediate risk, and high risk groups, if you look at the summaries, they actually all favor statin use, meaning that individuals in those groups who got statins did better compared to individuals that got placebo. And actually, if anything, the trend is individuals at high risk had a greater relative risk reduction than individuals who were at intermediate or low risk. And we've actually gone on and replicated this in a different study. There should be an arrow here. High genetic risk. So I showed you that there's perhaps a slight increase in relative risk reduction. But the bigger bang for the buck, I think, is that you get a greater absolute risk reduction. And the reason is that if you're at high genetic risk, you start at higher risk. And so if you lower that risk, you actually have a greater absolute risk reduction. And so these are the two primary prevention trials. And you can see that if you're at low genetic risk, intermediate, or high, individuals at high genetic risk have the highest absolute risk reduction. Again, because they're starting at the highest absolute risk. And this is an important statistic in clinical trials. When we think about the number needed to treat. So the number needed to treat is the number of individuals you need to treat with a therapy over a given time to reduce one event. And this is the number needed to treat is a reciprocal of the absolute risk reduction. And so if we take one of these trials, the ASCOT trial, if you're in the low genetic risk group, we need to treat 100 people over the course of 7 years to prevent one event. But if you're in the high genetic risk group, we can cut that in a third. And number needed to treat of 33 is actually quite reasonable in terms of thinking about a very safe and inexpensive therapy. So in conclusion, hopefully I've convinced you that in terms of precision genetics for rare diseases, we have sequencing-based testing that's in practice today. So I didn't talk about the gene panels for the different conditions, but they were alluded to earlier. That this is already providing precision diagnoses. We can actually do better than that too in gene-negative families. And so I think in informative families, we have the ability to do sequencing-based discovery. And there are now emerging genomic technologies and techniques that can provide a rapid path toward figuring out mechanism. In the common disease space, there are hundreds, potentially even thousands, depending on how these studies move, of markers across the genome that are collectively informative for risk. That polygenic risk scores have the ability to identify those who benefit most from prevention, at least in coronary disease. Despite that, the role in preventive medicine is to be determined. So there's all sorts of legal and ethical questions, but I hope that at some point we're going to be able to use this to move precision medicine forward. With that, I'll show you the view of my side of the state, which is down south on the other side of the river, thanks to my lab and everybody else that we collaborate with, and look forward to the panel discussion later.